Hello, everybody, and welcome back to Creation Myths, now broadcasting from my basement. Today's creation myth is created heterozygosity, and let me tell you, I have been looking forward to doing this one for a long time. Before we get into the nitty-gritty of this creation myth, I want to just make a quick note here. We're going to be doing this entire thing using creationist assumptions. I'm going to quote extensively from the book Replacing Darwin by Nathaniel Jeanson, because he leans very heavily on this created heterozygosity argument in that book. So we're going to get the argument straight from Nathaniel Jeanson in his own words, and when we show why that argument is wrong, we're going to use his own assumptions in doing so. So creationists, after watching this video and seeing why this argument is completely wrong, the last thing I want to hear is that I'm using evolutionary assumptions and not actually listening to the creationists. Everybody clear on that? Great, let's get to it. Now, this topic is a little bit technical, so we have to just establish a little bit of a foundation before we get to why created heterozygosity is wrong. So let's start with a look at genotypes and variation. The word genotype just refers to the genetic makeup of an individual at a specific location in their genome. Basically, for any given gene that you have one copy from mom and one copy from dad. That's called being diploid. And there are different versions of genes that exist. Those variants of each gene are called alleles. We have words for when you have the same alleles for a particular gene or when you have different alleles. When an individual has two alleles that are the same, for example, capital A, or lowercase a, that is called homozygous. And when you have alleles that are different for a particular gene, like capital A from mom and lowercase a from dad, that is called being heterozygous for that particular gene. Typically, when we use the words homo and heterozygous, we're talking about individual genes. Now, if you think back to your introductory biology class or watch my Evolution for Creationists series, you probably know that Gregor Mendel and his pea plants, he looked at genes that have just two alleles, a dominant and a recessive. And that's the case for some genes, but some human genes have dozens, hundreds, or even thousands of different alleles. The question that creationists need to address here is, how do we get all of this variation in about 6,000 years since creation? And I am addressing this kind of as an anti-young Earth creation argument, because it is young Earth creationism that employs the created heterozygosity argument. So what we need to see is, how do we get this variation in about 6,000 years? The answer is called created heterozygosity, the idea that Adam and Eve and the other created kinds were heterozygous, highly heterozygous, meaning that across their genome, instead of Adam having two of the same alleles for each gene, he had two different alleles for each gene, and the same thing for Eve, meaning you can get up to 4 different alleles per gene in your original created pair. And here we see this just very simply. We've got one individual, 2 different things, another individual, 2 different things. Now, what we talked about is that you have more than 4 variants for many human genes. Well, how do you get that? You get that through recombination. So through the process of crossing over during meiosis, which you maybe learned about if you took an intro bio class at some point, you get mixing and matching of your different genes, so you end up with many new variants over generations. This is how you go from your 4 created variants to all of the diversity we see today. That's the creationist argument. Like I said, I want to be very fair about how I'm presenting this argument, so I'm going to quote extensively from a 2016 publication by Dr. Nathaniel Jeanson and Dr. Jason Lisle called On the Origin of Eukaryotic Species Genotypic and Phenotypic Diversity, in which they elaborate in detail of how this created heterozygosity mechanism works. And they call it the CHNP hypothesis. That's created heterozygosity and natural processes. They write, the CHNP hypothesis proposes that diploid individuals were created heterozygous and that natural processes since this event, including recombination, gene conversion, mutation, and natural selection, etc., have distributed and or added to the original created genetic diversity, thus producing the genotypic and, consequently, phenotypic diversity we observe today. I'm going to keep going because I want to make sure we're clear on all of this. They write, to be sure, this is not a deistic hypothesis. Under the CHNP model, God doesn't create and then abandon his creation. Rather, the CHNP model recognizes that God is actively involved in his creation, providentially upholding it to this day, And the model recognizes that God works via means, including via the environment and the natural processes that he supernaturally designed and upholds. They continue. As an additional point of clarification, our CHNP model does not reject the operation of mutations, transposition events, or the like. Instead, we propose that kinds started with heterozygous genomes and that the genetic variety in these genomes was modified not only by recombination and other reshuffling processes, but also by mutation processes, only at rates consistent with documented genetic processes and parameters. Now, I know that was a lot, so thank you for indulging me having that 3 paragraphs worth of quoting of Jeanson and Lyle explaining this mechanism. Let's summarize the argument they're making. The explanation for existing genetic diversity is created heterozygosity and natural processes. The created genomes highly heterozygous, and then the existing genetic diversity is due to that heterozygosity, plus subsequent recombination events and mutation and gene conversion and all that other stuff. Plus, it's important to note ongoing divine involvement in these processes. Let's not leave that out. So now let's talk about why creationists are wrong. Now for the fun part. The first reason creationists are wrong is that you can't just make stuff up. Creationists are assuming an unobserved, ongoing divine intervention in biological systems. Okay, you can't just presuppose that and expect people to take you seriously. Jeanson writes in 2017's Replacing Darwin, in short, the creation and design model predicts with equal force to evolutionary theory each of the biological evidences we examined, and talk about things like sequence similarity and nested hierarchies and that kind of stuff. Problem is, that's not true. Because evolutionary mechanisms that lead to those things are observable. We can watch them happen and document them. Created heterozygosity and the associated ongoing, in the present, divine intervention in these processes is unobservable and untestable. The second thing that's a problem here is that Jeanson repeatedly claims that his model makes predictions that are confirmed. The problem is that those predictions are coincidentally the same predictions as evolutionary theory, and those predictions he's making, like, oh, the creation model, the created heterozygosity model predicts nested hierarchies. In 2017, when Jeanson wrote this book, or when Jeanson published this book, the nested hierarchies had already been discovered. You can't predict the die is going to come up a 6 after it's come up a 6. It's not how this works. So Jeanson's just wrong in terms of the explanatory power here and how his model is equivalent to evolutionary theory. That being said, the big problem with the created heterozygosity and natural processes model is that there simply isn't enough variation to explain the extant variation today. So you have the maximum number of alleles in 2 diploid individuals is 4, right? Adam's got 2 of each gene, Eve's got 2 of each gene, so you can have a maximum of 4 variants. Well, there are human genes with way more than 4 alleles, and the one we're going to talk about as an example of why this hypothesis doesn't work is the ABO blood group. As you probably know, there are 3 main blood types, A, B, and O. Well, that gene within those 3 major variants, there are over 1,000 different alleles of that gene. So there's lots of variants of the A, the B, and the O versions. Well, how do you get 1,000 variants from 4 initial alleles? In a young Earth timeline, you can't. It's simply not possible. Now, the answer that creationists give to solve this problem is recombination. Jeanson, in the book Replacing Darwin, writes that 99.6% of extant biodiversity is due to created heterozygosity. In other words, you start with your variation and then you have recombination, and that explains all but 0.4% of existing variation. The rest is explained by things like mutation. The problem with what Jeanson does here is he uses heterozygosity in a non-standard context. He writes, the term heterozygosity can be applied to a chromosomal pair at multiple levels of comparison. It can refer to a state in which every single base pair on one member of the chromosome pair is different from every single base pair on the other member of the chromosome pair. It can also refer to a state in which all base pairs but one are identical between 2 members of a chromosome pair. Heterozygosity simply refers to the state of being genetically different. He also writes, calculations on the theoretical combinatorial diversity from a given set of chromosomes are best done using formulas for combinations rather than permutations. That emphasis is mine. That right there is really important, that he's saying combinations rather than permutations. He also writes a little bit later, since chromosomes encode visible traits, these results imply that heterozygous ancestors also contain massive potential for diversity in visible traits. In other words, phenotypes. So he's saying you have two chromosomes that are very different from each other. You can mix and match them in lots of different combinations, and that leads to the potential for lots of different phenotypes down the road. The problem here is that this is a bait and switch. Jensen is distracting from what the actual problem is. He's talking about recombination between genes. So you have gene A over here and gene B over there, and you get different combinations of alleles between those two genes. But what we need to explain is variation within genes. How do you get all of the existing diversity within a specific gene? The problem for Jensen is that intragenic recombination, which the regions in red here indicate genes on this little chromosome, the regions in red are genes. Intragenic recombination with breakpoints within a gene, is far less common than intergenic recombination with breakpoints between genes. And then in blue in this figure, we have the exons, the actual amino acid encoding regions of those genes, in contrast to the non-coding introns. Well, intraexonic recombination is rarer still, and you need recombination within the exons in order to get all the variation that we observe. Now, this is all very general, so let's see what happens when we put actual numbers to this problem. Warning, we are going to do math. If you read Nathaniel Jensen's book, Replacing Darwin, which I strongly recommend you do, in this part of the book, when he talks about the potential for diversity from created heterozygosity and recombination, he just kind of skirts over the actual math here. But guess what? We're not going to skirt over the actual math. We're actually going to do the math and see why Jensen is wrong. So let's start with the number of alleles. And I want to be very clear here. We are going to use Jensen's assumptions, and we are actually going to be extremely generous. We're going to make assumptions that are more friendly to his case than the numbers he actually provides. So we're going to assume a 15-year average generation time, which is insane, but that's what we're going to assume. We're going to see that this yields 40,000 recombinations, and we're rounding up there. Jensen's numbers are actually 39,000 something, but we're going to say 40,000 recombination events per lineage. That works out to about 860 recombination events per chromosome. We're going to round that up to 1,000. To be clear here, we're assuming an equal distribution of recombination events across chromosomes, which is not the case. Chromosomes vary in size, and obviously the bigger chromosomes are going to have more recombination. But we're just going to equalize it across the genome. We're also going to assume an equal likelihood of recombinations at any point within a chromosome. That is also wrong, and I'll go into that in a little more detail later on. Finally, we're going to do some rounding, like we did right here, to make the math work better for Jensen. So at every point here in our math, we are making assumptions that are friendly to creationists, and it still comes nowhere near work. So let's see if we can generate the diversity in the ABO gene. As I said, there are over 1,000 variants in that particular gene in all of humanity. And that gene is about 25 kb long. That's 25,000 nucleotides. It's located on chromosome 9, which is about 140 megabases, or 140 million nucleotides long. That means this gene represents about 1/5,600th of that particular chromosome. Now, if we assume each recombination event yields two new alleles, because you have two existing alleles, and then they recombine, let's say those are now two new alleles. That's not always going to be the case, but let's assume that's going to happen. To get your 1,000 variants, we'll round down to 1,000 to make the math friendly for creationists. That means you need about 500 recombination events within that gene. And this is assuming 4 starting unique alleles at creation. That means you need 498 recombination events within that gene. We'll say about 500. Now, if we assume all recombination locations along chromosome 9 are equally likely, that means because this gene represents just 1/5,600th of that chromosome, we need to multiply our 500 recombination events within that gene by 5,600 to get the full number for that chromosome that we would need in order to accumulate the 500, and that works out to 2.8 million recombination events in chromosome 9. In Replacing Darwin, Jensen estimates there are about 1,000 events, 1,000 recombination events per chromosome per lineage since creation. So if we take that number at face value and assume 1,000 events per chromosome per lineage, that requires 2,800 distinct human lineages radiating from the arc. Did that many lineages radiate from the ark or pop into existence immediately after? No, that is insane. That makes no sense. We can also look at this with real numbers, though. Rather than using entirely gene-sense numbers, we know how frequent recombination is. So we can use those numbers and do more math. So for example, we know that there's about 1 crossover per chromosome per generation in humans. If we require 500 events within this gene, again, multiply that by 5,600, that means we require 2.8 million generations to accumulate those 500 recombination events within this one gene to get the diversity that we need. So at 15 years per generation and 6,000 years, that's 400 generations per lineage, meaning we need 7,000 lineages to accumulate the amount of diversity via recombination that we see in extant human populations. And I want to be very clear about how insane those assumptions are. 15 years average per generation and 6,000 years? No, it should be 4,000 because what really matters is what happens after the arc bottleneck. But we're just going to ignore that. And it gets worse for creationists. Recombination is not equally distributed. Most recombination events occur in less than 10% of the genome. These are called hotspots. These patterns are conserved across human lineages, which makes the problem worse for creationists by a factor of 10 to 100. So it's not 7,000 lineages you need, it's 70,000 or 700,000. The obvious conclusion is that you can't get enough diversity in a young Earth time frame, even with assumptions that are extremely generous to the creationist position. Now, this does lead to another interesting question. How long would it take to get this diversity? Well, that math has been done. The technique is called time to most recent 4 alleles, rather than time to most recent common ancestor, because we want to go back to this created heterozygosity starting state. So you assume created heterozygosity, look at the present diversity, the rates of recombination and mutation, and you work backwards to see how long it would take to converge at 4 alleles. Now, I want to be clear, this math that I'm showing is not specifically for the ABO gene. This is just in general for the human genome. This work indicates that the convergence time, the time to most recent 4 alleles, it's not 6,000 years, it's not 10,000 years, it's about 500,000 years. That's the median value. So you can see right here, these graphs both represent the distribution of outcomes. The median is about 500,000 years, or 495,000. If you want to use the mean, that's 461,000 years. Needless to say, that is incompatible with the young Earth timeline. There are a number of rescue devices that creationists use to try to make this hypothesis work. One of them is, well, maybe we can generate more diversity as these lineages radiate around the world. So they'll invoke mechanisms like drift, inbreeding, and adaptation to harsh environments. And this figure is one I've shown before. This shows the radiation of humans from the ark. The problem here is that these mechanisms reduce diversity and you end up with fewer alleles rather than more, so it makes the problem worse. Another fallback is, but mutations explain it. Well, GeneSid says almost all diversity is created, but putting that aside, other creationists will invoke mutations. The problem is that creationists generally claim mutations are virtually all harmful or destroy function, even beneficial mutations. So for a rundown of that argument, you could see Darwin Devolves by Michael B., for example. In order to explain extant diversity, mutations must generate thousands and thousands of new functional alleles. It still takes way too long to cram into a young Earth timeline, but if creationists want to make that argument that mutations are suddenly capable of generating all this new allelic diversity, great, fine. Thank you for acknowledging that that is possible. Finally, some creationists are just confused about things like basic population genetics. So, for example, I've heard the argument that drift and inbreeding in a lineage will cause hypermutation, and all of those new mutations then generate the diversity to get what we see today. Now, to be clear, that is wrong. The other interesting side of this is that that argument is used, for example, to talk about how Neanderthals got so different from Homo sapiens and why they seem to have a lot of genetic disorders. So that hypermutation is generating a lot of problems, but in the context of human diversity that we need to generate, it's generating lots of new functional alleles. So there's a contradiction there. But putting that aside, what happens with inbreeding is not that you're generating new harmful alleles. What happens is that you're increasing homozygosity, not the mutation rate. And when you're more homozygous within a population, you see more recessive phenotypes, and many of those tend to be harmful. This pedigree shows how this can happen. So you have two grandparents in this case, one of whom is heterozygous for a recessive disease-causing allele. Two of the siblings, each of whom are heterozygous, mate with each other, and you end up with offspring that is homozygous recessive. This offspring now has a new recessive genetic disorder that was not present in anyone else in this family tree. That's not a new mutation that resulted in that phenotype. It's simply the association of those recessive alleles with each other through inbreeding. So through inbreeding, you're generating more homozygosity. So on this argument, creationists are just wrong about basic population genetics. So to summarize the creationist position, creationists invoke created heterozygosity to explain extant allelic diversity in humans and other species. This is based on unobserved and unobservable assumptions. It is unable to explain extant diversity. There's simply too little variation at the beginning, and it takes too long to achieve the observed variation within a young Earth time frame. They have various rescue devices to try to fix this hypothesis, but those are insufficient, and some of them even make the problem worse. So, created heterozygosity? That is a big fat creation myth, and one of my favorites because it just makes no sense. As I've said before, if any creationists take issue with anything I have said here and you want to come on and correct the record, you are welcome to do so. Email me. My email is down below. You can come on and you can have as much time as you want to make your case refuting the arguments I have made right here. So thank you for watching, everybody. If you're enjoying what I'm doing, please like, please subscribe, please share it, and there are a couple links down below if you want to contribute to help me do more of this down the road. This has been the myth of created heterozygosity. Thank you, and don't get fooled.